So the next speaker is Jonathan Esensten, and Jonathan Esensten leads an experimental cellular therapy process development and manufacturing group at UCSF, which is focused on improving techniques for manufacturing T-cell-based therapies for patients with autoimmune diseases, solid organ transplant, and cancer. He was a Leukemia Lymphoma Society Fellow in Wendell Lim's lab and completed his residency in clinical pathology and fellowship in transfusion medicine and blood banking at UCSF as well. He's co-director of the Clinical Regulatory T-cell Manufacturing Group at UCSF and medical director of the UCSF HICTF and GMP facility, and a major contributor to the Institute's work to develop cell therapies for clinical trials.
Great. Thank you so much, Stacy, for the introduction. And thank you, Tom, for this incredible introduction to our program and the T-Reg therapy that we're doing. So in the next 20 minutes or so, I'm going to tell a story about another kind of T-Reg cell therapy. And this is a gene-edited T-Reg product that we are developing for a patient and a small family of patients with a rare disease caused by deficiency in IL-2RA or CD25, as most of us know, the protein. So a very quick background on T-Regs. Tom already gave quite a lot. So they're a small subset of CD4 positive T cells, and that actually makes it challenging to obtain them from the peripheral blood to manufacture them. There are only 5 to 10% of CD4s. So one of the challenges that we face in our manufacturing is isolating these Tregs to a high enough purity so that we can adequately manufacture them. And Tregs have multiple mechanisms of action, but one mechanism that I want to focus on, and that is going to be important and relevant for the particular disease that we'll be focusing on today, is deprivation of IL-2. So Tregs have constitutive expression of CD25 or IL-2 receptor alpha, and that allows them in inflamed tissues, even in non-inflamed tissues, to selectively bind up IL-2 and keep that IL-2 away from effector cells that may have lower expression of CD25. So just a quick review of how the IL-2 receptor works. So there are two components required for signaling: IL-2 receptor beta and IL-2 receptor gamma. The gamma chain is the common gamma chain shared with other cytokine receptors. And that has an intermediate affinity for IL-2 on its own. Most T cells, unless they're recently activated, are going to express just the beta and the gamma chain. But when T cells upregulate the alpha chain, and as again I said, Tregs have constitutive activation of the alpha chain, they have a very high affinity for IL-2, so 2 orders of magnitude higher, and therefore can compete for it in an environment where IL-2 is limiting. We know from work in animal models that IL-2 receptor alpha, or CD25, I'm going to use both terms interchangeably, is required for Treg function. And in the absence of CD25, Tregs don't work well, and these animals develop autoimmunity. So this is a study that looked at a Treg conditional CD25 knockout. On the top line here, you can see various different tissues that were sampled. They all show significant inflammation. These animals have autoimmune hemolytic anemia, weight loss from their inflammation in various organs, and die very quickly. There's a very similar type in a germline knockout of CD25. Of course, in those animals, it's both the Tregs and the effector cells that lack this expression of this protein. However, it seems that the Treg phenotype, the inflammatory phenotype, tends to predominate. So now I'm going to get into a clinical case that really kicked off this particular line of work, and it was a case of neonatal type 1 diabetes. The patient was born at term, but at a young age, 15 weeks, developed signs of type 1 diabetes, presented in DKA, was found to have the typical antibodies found in type 1 diabetes by 6 months of age. Because of the rarity of neonatal type 1 diabetes, this disease usually presents in older ages in its sporadic form. The patient was sequenced, a full exome sequence, and was found to have compound heterozygous mutations in IL-2 receptor alpha. And I should say that it was Kevin Harold at Yale, who was a longtime collaborator for our Treg program, who brought this patient to our attention and really handed it off to Alex Marson's group to do further work on it to see how CRISPR might be used to address this genetic defect. So a little bit more on what the genetic defect is. So no consanguinity, two totally different mutations, one of which causes a premature stop codon, one of which causes essentially a frame shift and a run-on in the protein. But in the end, CD25 is not expressed on T cells or Tregs in these patients. Okay. And so the name for the clinical syndrome caused by a lack of IL-2RA or CD25 is Ipex-like syndrome. So Ipex is the classic FOXP3 mutation causing defects in Treg function or stability. And Ipex-like is related Treg disease, Treg-driven disease, but it's caused by other mutations, most notably in IL-2RA, like in this case, STAT5B, or in other genes. And the phenotype can be quite variable in patients. FOXP3 mutations and Ipex usually presents very early in life and requires allogeneic stem cell transplant, whereas Ipex-like syndrome has a more variable and sometimes a more benign course. So in this particular family, here's the index case here. I'm pointing to with the arrow, who presented with type 1 diabetes. Turns out of the 5 SIBs, 2 other siblings were also found to have these compound heterozygous mutations. One of the SIBs was found to have autoantibodies, anti-islet autoantibodies, but no overt disease. And then the oldest patient, who we will be focusing on for the rest of this talk, developed ITP, autoimmune neutropenia, and still does have, to this day, chronic pancytopenia as the primary clinical manifestation of her disease. And as you can see from the flow plot below, looking at expression in CD4s of IL-2RA versus CD127, where we generally see the Tregs sticking up here as CD25 positive and CD27 low to intermediate. We see these Tregs are present in healthy donors, they're present in parents, they're present in the SIBs who have just one copy of the mutated allele, but we do not see these cells in the three patients with the compound heterozygous mutations. Now, they do have FOXP3 positive cells that do have the epigenetic profile that is typical of Tregs, but they appear to be non-functional, and they, again, don't have this typical flow profile. So our goal here was to try to find a way to genetically correct these patients' Tregs, since we need some preclinical justification. So in IL-2RA null mice, there is this autoimmune disease, and it's been shown by other groups that infusion of or adoptive transfer of Tregs into these mice very quickly normalizes levels of IL-2. So IL-2 tends to be high in these animals, since there's no sink to soak up the IL-2. And it also prevents clinical signs of autoimmune disease, which I mentioned earlier are prominent in these animals. And I've listed here the frequency of different sorts of autoimmune phenomena that are seen in these animals. So our goal here is to manufacture a gene-corrected autologous Treg product. We're going to call it GCT Reg from now on, to treat at least one of these patients, and perhaps the other 2 patients with IL-2RA deficiency, CD25 deficiency. And the approach we're going to take uses CRISPR-Cas9 technology. As I mentioned, since these patients do have Tregs, they just appear to be non-functional, don't express CD25. What we need to do is we need to somehow obtain those Tregs, purify them, and then using a Cas9 guide RNA, an RNP, to generate a double strand break at one of the mutation sites. And we picked the one of the mutations as the one we're going for here, along with a short DNA template to provide a corrected sequence, as well as a mutation in the PAM site so that the Cas9 will cut only once and not cut once the repair has been made. Putting all this together, our goal is to produce a population of autologous Tregs that have a correction in one of those two mutated alleles, which will, we hope, lead to re-expression of CD25. So this experiment was actually done by Theo Roth, who published, was in Alex's lab, published a paper a few years ago and looked at cells, Tregs from the three patients with the compound heterozygous mutations. And he showed that indeed, using this general approach, he could get re-expression of CD25 in a subset of Tregs from these patients. So in terms of proof of concept here, in a research lab scale, it does appear to work. Okay, but now the challenge is to develop a GMP compatible process to manufacture these. And whenever you move from a research lab scale, to a GMP clinical scale, lots of things are going to change. Really what changes is the context in which you're doing the work. So translational cell therapy requires, of course, the discovery, investigator-driven preclinical work, which is what Alex and Theo did so beautifully. But then we have to move into actual process development. How are we going to make this drug? It is a drug regulated by the FDA as such. And how are we going to manufacture that drug under cGMP regulations? And of course, along with that, we have a quality program that has to be developed, FDA filings that have to be done, a trial design that has to be developed alongside of the product. And all these pieces have to fit together in a harmonious way, and all has to be tracked along so that we get to our desired outcome, which is infusion of our patients. And so Tong already introduced the Treg group, so I'm not going to spend a lot of time, but just to say the work we're doing here is in the context of a group founded by Jeff Bluestone, co-led by me and Tong, that has long experience in Treg therapies, autologous Treg and now allogeneic Treg therapies for the past 10 plus years, since 2011. And again, a summary of the trials that have been run or are running out of our group right now for a variety of different indications, including autoimmunity and transplant. So how are we going to do this? How are we going to make gene-corrected Treg under cGMP? First, I'm going to define what is cGMP, because I think this causes a lot of confusion. So sometimes people ask me, does cGMP mean that you make the cell therapy product in a really clean environment, in a clean room? And that is true. We do this work inside of clean rooms, but the clean room isn't the magic of GMP. GMP has a whole lot to do with the quality of the materials that are used, the people who actually do the manufacturing, the qualification and validation processes that you go through, and the documentation of all steps. Okay, and of course, the quality program, an independent quality program that ensures that we're following all the rules, is an essential part of this. So you can't test quality into a product. You can't make something in a research lab and test it and say, oh, that's good enough to give to a patient. You really have to design this cell therapy to have quality from the beginning, the quality that you need to convince the FDA that it is a good idea to proceed with a clinical trial. So I'm going to talk briefly about how we obtained the materials for this manufacturing process. So these are some, but not all, of the critical materials. And each one of them, we had to find a vendor that was willing and able to give us a high enough quality material so that we could justify using it. So Cas9 protein now is being made in GMP grade. as our guide RNAs and the single-stranded DNA repair template that we're using. So all of these are being made at either GMP or a GMP-like, which is mostly a semantic issue, but less expensive and probably just about as good quality level as GMP. We also need sorting antibodies because, as I mentioned, Tregs are a very minor component of all CD4s, so we have to be able to identify them. And then cytokine IL-2, CD4 microbeads, because we do a CD4 selection, and in this case, using rapamycin to prevent outgrowth of non-Treg cells. So our general Treg manufacturing process requires the expression of CD25 to isolate the Tregs and to separate them out from all the non-Treg cells. Because if you put Tregs and non-Tregs together in a culture and stimulate the cells, the non-Tregs will overtake the culture and you'll have very few Tregs. So we really have to get a relatively pure population. And so this is the kind of flow plot we see, and we do droplet-based sorting to get the Tregs. Now, if you don't have CD25, this really becomes impossible to separate the Tregs and the non-Tregs to a high purity by flow, okay? So we had to develop an alternative manufacturing strategy for these cTregs, of course, which do not express CD25. And so this is what we came up with. And when I say we, I really mean Brian Shai, who was a resident and then a fellow, and now just recently joined the faculty in Department of Lab Medicine, has continued to push this forward. So this is the approach that he came up with to sort Tregs to low purity, relatively low purity at the beginning, based on two markers, CD4 and CD127 low, and then expand those Tregs under the cover of rapamycin, which, as I mentioned, decreases the proliferation of the effector T cells while still allowing some proliferation of the Tregs, and has been used extensively by other groups in Treg manufacturing, although not ours until now. And then we gene correct the Tregs by electroporating in the Cas9 RNP and the template, the DNA template. And then we do a second sort, droplet-based sort. And this time we can sort using CD25 because CD25 now has been corrected, at least in one allele, and then re-expressed in the Tregs. Now, of course, you might be thinking, well, isn't that a problem? Because if you stimulate T cells, they're all going to upregulate CD25. So how are you going to tell the difference between the Tregs and the non-Tregs? So that is an issue. And so what we did is we had to figure out how long it takes after a stim for the Tregs and the non-Treg cells to essentially separate out by CD25 expression. And what we found is essentially by day 9 here, but even more so by day 14, we get a separation where cells that were totally impossible to distinguish soon after a stim here on day 1 by CD25 and CD127 expression become much more separable. So the Tregs here are in red and the effector cells are in blue. And so we can imagine doing a second sort and grabbing as many of those Tregs as we can. So we did a clinical scale manufacturing run in our GMP facility with all of the high quality reagents that I mentioned, with all of the standard operating procedures, with the trained personnel. We did it all at scale with a healthy donor leukopak because we wanted to test this out on a healthy donor leukopak before we went ahead and got patient cells to do it. And so this is a very brief overview of what that process looked like with some of the cell numbers involved. And the end result was very encouraging, that at the end of our expansion process, we got about 200 million Tregs, purity about 94%, all of which were gene corrected for CD25. Of course, they started out expressing CD25 because they're from a healthy donor, but I'll show you data in a moment that we think that the cells that did not get the correction actually didn't proliferate in the culture because they did not have CD25, and so we're at a disadvantage in competing for IL-2. So this was the critical second sort, so the sort that we do after the correction. And what you can see is that at the beginning of the day, we have about 54% FOXP3 positive cells in the culture, so about half Tregs and half non-Tregs. And you can see there's a spectrum of CD25 expression. So most of the cells that are CD25 low here are not cells that have had CD25 deleted, although that could be it, but probably effector cells that have just downregulated CD25 because it's been a while since the initial stimulation. And then after the sort, we find that we get a relatively high purity, about 86% by FOXP3, all of which are expressing CD25. And we think this is probably good enough for the purposes of our clinical product. Our goal is 70%, which is something we've discussed already with FDA and we'll continue to discuss what the actual target has to be. And here's the final product purity from our initial GMP scale manufacturing run, where the final GCT Reg FOXP3 positive percentage is 94, and some control Tregs here on the right. You can see looks very different from non-Tregs on the left. And we did Sanger sequencing to look at these final products, and what we found was by Sanger sequencing, which of course doesn't have the highest sensitivity, but gives us a pretty good view of what the majority of the cells have going on. We find that we have a mutation in the PAM site, which is part of the design of the repair template to prevent cutting after the repair. And then we see the correction at the mutation. So it's a very, very clean correction in these cells at the end of manufacturing. So conclusion, so we completed an initial qualification of a GMP compatible manufacturing process for IL-2RA gene-corrected Tregs. We have ongoing pharmacology and toxicology studies to satisfy FDA requests, and these will be required for the IND package. A multidisciplinary team has evaluated the oldest patient in this family and has endorsed GCT Reg as a viable therapeutic option when looking at all the other clinical options, the patient's clinical history, and the patient herself has expressed a desired willingness to proceed on with this if the IND is accepted by FDA. So our plan is to administer the GCT Reg as soon as we get go-ahead from FDA for a single patient IND. And if all goes well, of course, there are two other siblings who, depending on their clinical status, may also be candidates for this very much personalized therapy targeting a rare mutation in a rare disease. So with that, we are at the hour, and I'm happy to take any questions, acknowledgements. This is a huge group that's been working on this for many, many years and funded by from a variety of different sources. I particularly want to give credit to Alex Marson, Kevin Harold, Brian Shai, and Alvin Ha, who have been really instrumental in pushing this GCT Reg project forward.